chromi is an experimental collaboration between designers and scientists working in synthetic biology. In 2009, 7 Cambridge University undergraduates spent the summer learning the tools of synthetic biology, which is essentially a new approach to genetic engineering. Using standardized sequences of DNA in a format that's called biobricks, they learned to engineer bacteria. They designed their own biobricks using genes copied from existing organisms, inserted them into E. coli, and created bacteria that secrete colours visible to the naked eye. E. chromai went on to win the International Genetically Engineered Machine competition at MIT in 2009.
And joining us now is one of the winners of the iGEM competition. Welcome to Science Friday, Ms. Mullen.
Hi, thank you so much. I'm part of the Cambridge 2009 iGEM team, and our project was called E. chromai. And what we were trying to do is to improve bacterial biosensors. They're bacteria that can tell you the concentration of a pollutant in water. And they can do this because inside them they have a detector. So we developed 2 different parts, the sensitivity tuner, and this actually tells the detector when to turn on and when to turn off. So you have control over what level of the pollutant you're detecting.
And how does the bacteria show that it's on or off?
We use something called a color generator, which means that our bacteria changed color when the detector got switched on.
Wow, so they light up in a different color.
They actually change color visible to the naked eye.
So let's say if you put a swab of the bacteria in a polluted river, bacteria would just change color.
Yep, exactly. Though you'd probably want to put a sample of your water on a bacterial plate, maybe not the other way around.
Well, how would you envision something like this being used other ways in the future?
As designers, we worked with the team to explore E. chromai's potential as they were developing it in the lab. And together we imagined the timeline proposing ways that living colour could evolve over the next century. These scenarios, some of which are shown in this film, explore the different agendas that could shape E. chromai's use and in turn our everyday lives.
One of the first real applications for this technology may arrive quite soon. A cheap disposable biosensor for testing groundwater contaminated by arsenic.
Bacteria could also be used to produce natural colourings and dyes. By 2015, there may be a profession of people who hunt for new pigments in the genes responsible, bringing them back for use in the food and textile industry. By 2039, you can go to the supermarket and buy this simple probiotic yogurt for cheap personalised disease monitoring. The yogurt drink contains E. chromai bacteria, which establish a colony in your gut. They monitor for chemical signals that indicate the presence of a wide range of diseases. If they detect a disease, they start generating the corresponding coloured pigment, producing an easily visible output to prompt you to seek your doctor.
2049 sees the rise of the Orange Liberation Front, a terrorist organisation from the Netherlands who are angry because a biotech company in China has patented the gene for the colour orange.
In 2069, Google releases pollution mapping bacteria into the atmosphere that turn red in the presence of excess CO2. And as the saying goes, red sky in the morning, Google health warning.
Our collaboration meant that E. chromai was a technology that's designed from the start at both the genetic and the human scale, and with a long-term outlook. We found that design and science could have a meaningful exchange in the lab, which could prove useful when developing technologies in the future.
You know, I think it's a new term to most of the public, synthetic biology. We've heard about all kinds of genetic engineering, stem cell research, but I think the phrase synthetic biology has not been used a lot lately. But I guess we're going to be hearing more of it.